Okay, welcome everyone. Today I'll be walking you through the fascinating journey of how nucleotides and ribose are made into glucose, and how these components come together to form ATP, which is crucial for DNA replication. So what I'm showing you is the metabolic map poster that we have been making the past couple of weeks. And so we will start here. We're going to start with 2, with 7, and with 8 here for glycolysis. So we're going to start with glycolysis. This is the first step in glucose metabolism. Here, I'll go like this. It's the first step in glucose metabolism, where 1 molecule of glucose is converted into 2 molecules of pyruvate, and we result in a net gain of 2 ATP and 2 NADH. So during glycolysis, glucose is phosphorylated into glucose 6, and then this is further processed into our fructose. So we have the glycolysis here, into our fructose 1,6 phosphate pathway. And then this molecule is split into 2 and 3 carbon molecules, which eventually produce our pyruvate, our ATP, and our NADH. Okay, and so then from there, we are now going to be looking at this one and this one, so our 12 and 13. So from here, we have our Krebs cycle. So pyruvate will enter the mitochondria and will be converted into acetyl-CoA. So we see it entering the mitochondria. This combines with oxoacetate to form citrate, and then through a series of reactions here, we have our citrate synthesis. So we have citrate is transformed back into oxaloacetate, and we produce 3 NADH, 1 FADH2, and 1 GTP-ATP per acetyl-CoA molecule. So this cycle, so that's what we'll see here, is it transforming. This cycle is vital for generating high-energy electron characters that are used in the next step. So then the acetyl-CoA produced from B oxidation enters the citric acid cycle. So this acetyl-CoA will enter the citric acid cycle, and this also contributes to our production of NADH, FADH2, and then ATP. So this links our fatty acid degradation directly into our ATP production through the Krebs cycle. Next, we're going to be looking at these two here. So this is our electron transport chain, and then our ATP synthase. So in the electron transport chain, NADH and FADH donate electrons to complexes in the inner mitochondrial membrane. So as the electrons pass through these components, or these complexes, protons are pumped into the inner membrane space, and this creates a proton gradient. Protons then flow back into the mitochondrial mitochondrial matrix, and then that's through ATP synthase as shown here. And so this drives the synthase of ATP from ADP and inorganic phosphate, so PI. This process is known as oxidative phosphorylation and is the primary method of ATP production in cells. Next we're going to look here. So this is our PPP pathway. So to synthesize nucleotides and ribose, we turn to the pentose phosphate pathway. So this pathway here generates ribose 5-phosphate, which is essential for nucleotide synthesis, and NADPH, which is crucial for anabolic reactions. So glucose 6-phosphate from glycolysis is oxidized to ribulose 5-phosphate, which is then converted to ribose 5-phosphate. Okay, so the ribose 5-phosphate is a precursor for the nucleotide synthesis, and this reacts with nitrogenous bases like adenine to form nucleotides. So we have it reacting with adenine, and then this process ensures that we have the necessary building blocks that we need for DNA replication. Okay, so then we're going to be looking at these squares going down here. So to start, we're going to have ATP produced through the pathways that we've just discussed, and this is used as an energy source for various processes, including DNA replication. So like here you can see that during replication, the DNA helix will unwind the DNA double helix, and then DNA polymerase uses ATP to add nucleotides to the growing DNA strand, and then this provides the energy required for DNA synthesis to go through here. And so this is our metabolism poster pathway. So in conclusion, so starting from glucose and CO2, we produce ATP through glycolysis, and then through glycolysis, the citric acid cycle, and then the electron transport chain. And then ribose 5-phosphate from the pentose phosphate pathway is used to synthesize nucleotides, including ATP, and then this is essential for DNA replication and other cellular functions. So understanding these metabolic pathways highlights the complexity and efficiency of cellular energy production and its crucial role in maintaining life.